In this video, I'm going to talk about the process of urine formation. One of the most important things to remember about urine formation is that it is highly dependent on blood pressure. Right? So we're going to spend a lot of time looking at how blood pressure and the regulation of blood pressure is going to affect urine formation. So urine formation takes place in 3 steps. The first step is glomerular filtration. This is where both good and bad stuff will be filtered out of the blood plasma and then sent into the renal tubule. Then we have the process of tubular reabsorption and tubular secretion. These 2 processes happen simultaneously, but they're doing opposite things. The process of tubular reabsorption is taking some of the good stuff that got filtered out of the blood and putting it back into the blood. Tubular secretion is a second chance to take stuff that's bad and in the blood that didn't make its way into the filtrate and to actually put it into the renal tubule. So here it is, the big picture of urine formation. Looks a little bit scary. So we're not going to go into all of these massive details, but what we're seeing happening here are those processes that I mentioned. The filtration step, tubular reabsorption, and tubular secretion. So let's start at the beginning. We'll start with glomerular filtration. Okay. So during the process of glomerular filtration, water and dissolved substances are going to filter out of the leaky glomerular capillaries. Don't forget, so these are just blood vessels, but they're very, very leaky. Think of them as having lots of holes in them. Okay. So water and dissolved substances from the blood plasma are going to leave the glomerulus and enter into the space of the Bowman's capsule. So there are holes in the glomerulus that allow fluids and small substances to leave, but proteins and cells are much too big to leave, so the proteins in the cells are going to stay in the blood. The blood pressure in the glomerulus is what's forcing the fluid out of the leaky capillaries into the capsular space. But at the same time, there's an osmotic pressure being exerted by the plasma proteins in the glomerulus, and those plasma proteins are trying to draw fluid back into the glomerulus. So essentially the proteins that are remaining in the glomerular capillary are acting as an osmotic agent, which is drawing fluid back into the glomerulus. So basically we have 2 forces here. We're pushing fluid out of the glomerulus, but we're drawing some fluid back into the glomerulus. So who's going to win this battle? Well, the blood pressure is slightly higher than the osmotic pressure, so overall, fluid ends up leaving the glomerulus and entering into the space of the Bowman's capsule. The fluid that we form here is called filtrate. The glomerular filtration rate, or GFR, is the net result of these 2 opposing pressures in the renal corpuscle. So the filtrate that we form in the process of glomerular filtration consists of water and dissolved particles in the water. It's very similar in composition to blood plasma, with the exception of there aren't any proteins. Right? So the filtrate is going to contain water, electrolytes such as potassium and sodium. It's going to have acids and buffers. It's going to have glucose and amino acids, but it won't have proteins and it won't have cells. So now we're going to take a closer look at how we regulate glomerular filtration rate, because regulating glomerular filtration rate is one of the main ways that we're going to regulate what's happening in the kidneys. So anything that affects blood pressure or the osmotic concentration of blood can potentially alter glomerular filtration rate and affect your kidney function. So see if you can predict the effect on GFR if you increase blood flow and water pressure coming into the glomerulus. Right? Would you make more filtrate or less filtrate? If you said more filtrate, you're correct. If you kind of think of this as being like a garden hose, think of your glomerulus as being like a garden hose, but your garden hose has holes in the wall of the hose. So you got water squirting out of the little holes in the wall of this garden hose. If you increase the water pressure coming into the hose, then you're going to get more water squirting out of those tiny little holes. So essentially, this is how the glomerular capillary is going to work. As you increase the blood flow and the blood pressure in the glomerulus, you're going to form more filtrate. More fluid will leave through the holes in the wall of your glomerulus and enter into the capsular space. Can you predict now the effect on GFR if you increase the blood flow out of the glomerulus? Right? If you increase the blood flow out of the glomerulus, the pressure should drop in the glomerulus. And if the pressure drops in the glomerulus, you're going to have less leakage out of those holes, which means that you're going to lower the amount of filtrate that's formed. So let's take a look at these two different situations. Let's start by looking at the situation if glomerular filtration rate is low. So if your glomerular filtration rate is low, then basically what this means is that your blood is not being filtered. Wastes and acids are staying in your blood and in your body, and you can get sick from this. And the kidneys, thank goodness, are capable of detecting low glomerular filtration rate. And if they detect this low GFR, the kidneys can take action. So one of the things that the kidneys is going to do is that the arterioles leading to the glomerulus, those afferent arterioles, are going to dilate, which will increase the blood flow into the glomerulus, which will increase the pressure in the glomerulus, increasing your glomerular filtration rate. You're going to start squirting more fluid out of those holes, forming more filtrate. Another thing that the kidneys will do is they will secrete an enzyme called renin into your blood plasma. So renin is not a hormone, it's an enzyme. And the kidneys release the renin into your blood plasma. And once it's there, the renin will bump into something called angiotensinogen. And the renin converts angiotensinogen into something called angiotensin 1. So this is all happening in your bloodstream. The angiotensin 1 is then converted into something called angiotensin 2 in your blood plasma. Angiotensin 2 is the main product that we're trying to get here. Angiotensin 2 is considered a hormone, and it causes vasoconstriction of the efferent arteriole. So what this is saying is that the angiotensin 2 will eventually make its way to that outflowing arteriole of the glomerulus, and it's going to cause it to constrict. So basically, we're closing off the outflow of the glomerulus, which is going to cause the blood to back up in the glomerulus, increasing the pressure in the glomerulus, which will increase the filtration rate, increasing the GFR. So this was a roundabout way to also help to increase that low GFR to make it higher. Angiotensin 2 is also going to affect your brain. It's going to cause you to become thirsty, and it's going to which will cause you to seek water. And by taking in water, you're going to increase your blood volume, which increases your blood pressure, and increasing blood pressure will increase your GFR. Angiotensin 2 also causes the release of aldosterone and ADH hormones. So both of these hormones are going to cause the kidneys to pull water out of filtrate and put that water back into blood. And by putting the water back into blood will increase the blood volume, which will increase the blood pressure, which will increase the glomerular filtration rate. So all of these things together, all of them help to increase the glomerular filtration rate. So let's kind of review what happens when our glomerular filtration rate is low. So if our GFR is low, we need to increase it. How do we increase it? Number one, we dilate the afferent arteriole, which will bring more blood into the glomerulus, which increases the pressure there, increasing GFR. The kidneys release renin, which leads to the activation of angiotensin 2. Angiotensin 2 causes the efferent arteriole to constrict, which creates an increase in pressure in the glomerulus, increasing GFR. Angiotensin 2 also causes you to become thirsty and seek water, which will increase blood volume, blood pressure, and increase GFR. Angiotensin 2 also causes the release of aldosterone hormone and ADH hormone, and these two hormones will cause you to save water from your urine, which will increase your blood volume, increase your blood pressure, and increase your glomerular filtration rate. All right, but now let's look at the opposite situation. What if your GFR is too high? This is bad too. You don't want low GFR, you don't want high GFR. High GFR is bad because the high pressure inside the glomerulus can actually cause the glomerulus to basically blow up. Basically, it starts to disintegrate, right? And once you break down that glomerulus, you've lost a nephron, and if you continue to do this and continue to lose these nephrons, they're really bad at repair, you're going to start to have kidney failure. So the kidneys can detect this high GFR and they take action. One of the things that they do is they constrict the afferent arteriole. And by constricting the afferent arteriole, you have less blood flowing into the glomerulus, causing the pressure to drop there. So now you've reduced your risk of popping your glomerulus, right? This also will decrease your glomerular filtration rate. Another thing that's going to happen is we're going to dilate the efferent arteriole. By dilating the efferent arteriole, we're going to increase the blood flow out of the glomerulus, relieving that high pressure in the glomerulus. So you can see by constricting the afferent arteriole and dilating the efferent arteriole, both of these things lead to less blood pressure in the glomerulus, decreasing the glomerular filtration rate. So the process of urine formation had 3 steps: glomerular filtration, tubular reabsorption, and tubular secretion. So we've already looked at glomerular filtration. So now let's take a look at the last 2 parts, tubular reabsorption and tubular secretion. Okay. Once the filtrate is formed in the renal corpuscle, the filtrate then enters into the renal tubules, forming a new fluid that we call tubular fluid. Along the wall of the renal tubule, both tubular reabsorption and tubular secretion occur. These processes are going to take substances away from and add things to the tubular fluid. So now let's take a look at the process of tubular reabsorption. As I mentioned, tubular reabsorption and tubular secretion happen simultaneously. I could have easily started with tubular secretion, it wouldn't have mattered. Tubular reabsorption is looking at the process of where we take the good stuff from the tubular fluid and bring it back into the body before it leaves the body through the process of urination. So in the filtrate, we have a lot of good stuff like water, glucose, amino acids, vitamins, and some ions that we don't want to necessarily pee out. So we need to take it back into the body. And this is what happens during tubular reabsorption. So this is a process in which substances are transported from the tubular fluid across the wall of the renal tubule cell. So now the substance is inside the renal tubule cell. And then we have to get it out of the renal tubule cell and then into the extracellular fluid and then into a peritubular capillary. Okay. And so we're moving things across the cell membrane here, right? We have to move a substance across the cell membrane of the renal tubule cell to get it into the cell, but then to get it out of the cell and eventually into the blood, it has to cross a cell membrane again. So this does require transport across the cell membrane. So we're going to use transport mechanisms like active transport and facilitated diffusion. In the case of water, we'll use osmosis. So we use these processes to get the substances reabsorbed. This is mostly happening along the proximal convoluted tubule, but it does also happen along the nephron loop and the distal convoluted tubule. So as I said, during the process of tubular reabsorption, substances have to move out of the tubular fluid across the renal tubule cell into the extracellular fluid, or interstitial fluid if you will, and then into the peritubular capillary. Right? So let's take a look at that little green molecule there. That's supposed to be glucose. So we have glucose in the tubular fluid, right? But we don't want the glucose to end up in our urine. We worked really hard to absorb that glucose in our small intestines. And the last thing we want to do is pee it out in our urine. So we want to reabsorb it. So the glucose has to move into the renal tubule cell, then out of the renal tubule cell, into the interstitial fluid, and then into the peritubular capillary blood, and now we have reabsorbed our glucose. So we know tubular reabsorption has occurred when a substance is present in the filtrate but is not present in the urine. The process of tubular reabsorption does have a clinical application. If there is an excess amount of something in the blood plasma, there will be an excess amount of that substance in the filtrate. And if there's an excess amount, then likely the process of tubular reabsorption cannot occur fast enough to reabsorb all of that substance, and so that substance will then appear in the urine. An example of this is what happens during diabetes. So someone that is diabetic experiences glycosuria. So basically they have an excessive amount of glucose in their urine. Normally you should have no glucose in your urine, right? But during diabetes, there's a large amount of glucose in the bloodstream. So that means we end up with a large amount of glucose in the filtrate. And then we overwhelm our glucose reabsorption mechanism, so not all of the glucose ends up being absorbed and some of it ends up in the urine. Actually, back in the day, this is kind of gross, but back in the day, that's how they used to diagnose diabetes, was that sometimes the doctor would take a little sip of the urine and see if it tasted sweet from the glucose. Anyways, by the end of the proximal convoluted tubule, the following substances should have been reabsorbed. So about 100% of the filtered nutrients. So this is glucose, amino acids, vitamins, that sort of thing. 80 to 90% of our bicarbonate buffer should be reabsorbed. A little over half of the sodium and half the water, and about 50% of our chloride ions and potassium ions. Don't forget, water is reabsorbed through the process of osmosis, right? And water reabsorption is closely associated with the reabsorption of sodium and other electrolytes. And this is because, of course, water likes to follow solutes. So as you reabsorb things like sodium and possibly potassium, then the water will follow that sodium and potassium as it is reabsorbed. Any water that enters the collecting duct is only going to be reabsorbed in the presence of antidiuretic hormone. So in other words, any water that's in the collecting ducts will end up in our urine unless you happen to have ADH present. The antidiuretic hormone would then cause you to pull some of that water out of the collecting ducts and bring it back into the body. So tubular secretion is essentially the opposite of tubular reabsorption. With tubular secretion, we have like a second chance to take bad stuff that is still in the blood and put it into our tubular fluid. So sometimes the process of filtration is not perfect, and there are still bad things that are in the blood that need to be outside the body. So the process of tubular secretion is a way to take this bad stuff out of the blood and put it into the tubular fluid for excretion. This is a process in which substances will be transported from the peritubular blood plasma into the extracellular fluid, or if you will, interstitial fluid, then into a renal tubule cell, then out of the renal tubule cell into the tubular fluid. So it's the reverse direction of tubular reabsorption. Okay? Examples of substances that are secreted include penicillin, which is an antibiotic, hydrogen ions, in other words, acid, and then potassium ions. And most of these are going to be transported across the renal tubule cell membranes through active transport. So we are going to use ATP energy to move these substances into the renal tubule cells and then out of the renal tubule cell into the tubular fluid. So again, the process of secretion involves moving substances out of the peritubular blood into the extracellular fluid, into a renal tubule cell, out of the renal tubule cell, and then into the tubular fluid. Let's take a look at a specific example of secretion with penicillin, right? So penicillin is an antibiotic. This is what its chemical structure looks like, but we're just going to call it penicillin. Right? And we don't want penicillin to stay in the blood forever. Right? Penicillin is eventually cleared from the body through your kidneys. So the penicillin will leave your peritubular capillaries, and then it will be transported across your renal tubule cell membranes using active transporters. These active transporters are called OAT transporters. And now the penicillin is in your tubular fluid, and it can be excreted through the process of urination. So that's just our penicillin traveling with the rest of our tubular fluid down our collecting ducts, and eventually will be excreted outside the body. So let's take a look at another example. Here we have some acid in the tubular fluid. Is this acid going to be secreted, reabsorbed, or neither? Well, if the acid was actually in the peritubular blood, we could secrete it into the tubular fluid, but it's already in the tubular fluid, so we can't secrete it. So our other option is reabsorption. And do we really want to add acid to our blood? No. So really what's going to happen here is the acid is going to enter into the collecting ducts and eventually will be excreted outside the body.